First class. It is about metastasis and epithelial mesenchymal transition. The most important thing that you want to know about metastasis is that it is responsible for more than 90% of deaths in cancer patients. However, the sad truth is the mechanisms are poorly understood. If you remember, I told you about metastatic tropism, which means why from a particular primary tumor, why does it go to specific tissues as secondary sites? There are several theories, but we still have not understood the mechanisms very clearly. Again, that is very important because this is the major culprit for all the deaths. So, that's why it's important to study that kind of separately. Of course, the basics on metastasis I have already told you. There will be slight repetition, but it is justified because we are going to study some more details also together with some basics. So, sometimes you call them as secondary tumors because from the basic thing is it is a spread of tumor from its primary site to a distant site. So, another organ. 90% of the cancer deaths I already told you. So, the metastatic process requires anchorage independent tumor cells because you see they have to lift themselves up from the primary tumor and then they have to go to some other tissue. So, naturally they will degrade the extracellular matrix and they will enter the circulation either in blood or in lymph. So, that is the thing. So, ultimately the tumor goes from being local to systemic. Now, there is a new area that is emerging. Can you detect circulating tumor cells in the blood? So, that will be a non-invasive method of assessing whether you are going to have metastasis, right? Because if you have particular tumor cells, of course, it will not tell you where it is going to land up, but it may tell that some tumor cells are floating in your blood and they are likely to go and settle down in some location. So, you can be careful about it. So, that there are methods, people are working on this. This is one active area of research. People are looking for circulating tumor cells, but of course, they are not that common. They are very small in number and it is a rare phenomenon also, but it can be detected. The metastatic cells have to somehow colonize at their new area, and to regrow secondary tumor must activate another important process which we all know, angiogenesis. Some of the important events are shown here which we have already discussed, that is how the tumors are say slowly breaching the basement membrane and then getting into the blood cell which we call as intravasation, and then they have to come out of the blood. Either they can enter blood or they can enter the lymphatic system, and this is what we call as extravasation. And then their target sites which can be bone marrow, lymph nodes, lungs, any other tissues and so on. So, they have several avenues of destination, in the sense several important destiny awaits them. For example, some of them can die, cell death, or they can remain dormant, or they can do what is called dormant micrometastasis. Of course, the micrometastasis may slowly grow also, growing micrometastasis. These are described here. So, to begin with the local invasion, breakage of the basement membrane I told you, and then intravasation, extravasation, we have discussed all this. If they have lost the anchorage, they may also undergo apoptosis, that is cell death. They can undergo cell death, or they can be in a state of dormancy, either a single cells or a small collection of cells which we call as micrometastatic lesion that proliferated but could not recruit a vascular bed for further growth. So, that is why they remain small. Dormant cells can resume proliferation, which is what we call as growing micrometastasis. You remember, I have described, I have shown all these phenomena, but I have not described that. I showed only as a cartoon in the beginning. That is why this part is a little bit explanation of whatever we knew already. So, micrometastasis can enlarge if angiogenesis begins. So, angiogenesis is a very important thing to turn them into macroscopic tumors. So, colonization is a kind of rate limiting step in metastasis. So, to proceed further or not, that kind of thing is decided by this step. Macroscopic tumors must adapt to local conditions at the secondary site, that is very important. So, basically they want to regrow, then they to colonize in a secondary site and then becomes metastatic. So, relatively this is rare, and I also told you it is a rate limiting step. We have discussed some of the old theories, so called Paget's seed and soil hypothesis. Primary tumor cells spread randomly to all parts of the body via circulation and lymph, and few survive and adapt and proliferate in a new organ. Just like if you take seeds and spread it through this campus through helicopter, let us say, it will fall on a rock, means it is not going to grow. So, it depends on the place where it ultimately lands up. That is one theory. As I told you, this is definitely not a good theory for all cancers. It might be explaining some cancers, but really not suitable for everything. Modern view is that it is not random, it is usually found in certain specific target organs. Because we know the so called metastatic tropism, which I have already explained, only certain primary sites will land up in certain specific secondary sites, that we have already explained, and I have given you some examples also. The one example is colon cancer cells, they will go to liver due to portal vein circulation. In this case, you may say the Paget's seed and soil theory is good or something like that, because it is neighboring one anatomical location and all that, but it is not true for other things. For example, breast cancer can be ending up in brain, which is not a nearby organ. So, it will not work for everything. So, how to explain the organ specific metastasis? Circulating cells get preferentially trapped in certain organs. I told you they may act as a brake sometimes. They go and there are some adhesive molecules, so they get stuck up in that place, they circulate and they get stuck up due to certain adhesion molecules, and then in that place they may penetrate the blood vessels and get settled in the nearby tissues. That is one possibility. The other thing I have already told you, chemoattractant hypothesis. So, the receptors will be there, but the ligands will be present in certain tissues. So, they will go towards the ligand and settle down in those tissues where the ligands are present. So, that is the theory that is invocated for breast cancer. So, the ability to colonize is not fortunately is not a very frequent event, it is a rare event. So, that is good for us, because we do not want metastasis to occur. So, the more successful only targeting certain organs for metastasis. So, that is why there are about, this can explain in about 20% of the cases. Based on animal models, it is said that it is only about 0.01% of the cells that are freed from their primary location will actually progress to micrometastasis, which means more than 90% of them will not go to a secondary site, which is good, it is good. So, it is rare. Why? Because they have lost anchorage dependence. So, they must survive as suspended cells, because they have to, they cannot survive in the suspended situation, right. Of course, cancer cells can survive, but since they have lost the anchorage dependence, the possibility is less. That is one thing, it is very rare. One possibility is that the cells expressing certain receptors like integrin alpha 5 beta 3, they resist apoptosis. Integrins, this particular integrin signals via SARC to increase the cell survival, so that they can survive and get settled in other things, other tissues. Early stage of forming a viable micrometastasis is a rate limiting step. So, that is why the macroscopic tumors are also rare. It is not that it is very common, but it can still occur. Recent experiments, you see here, they have shown some experiments, show that nutrient starvation can induce autophagy. This is another phenomenon that is invoked in metastasis also. Autophagy that causes cancers to shrink and adapt a state of reversible dormancy, and cells may even resume proliferation when you get nutrients. Initially, there is a nutrient deprivation because they are going to a new environment. Maybe they are not getting the same nutrients as they used to get in their original site, primary site. So, they prefer to be in a state of dormancy. When they get the nutrients again, they may regrow. So, that is also noticed. So, apart from that, I already told you there are several signals which ultimately turn them into cancer cells. That is, so many hallmarks I have described already. There are inhibitory signals, there are apoptotic signals, there are many other signals, replicative signals and all that. One has to kind of, there are balancing forces. There are always, if you have apoptotic signals, there are anti-apoptotic signals. If there are replicative signals, there are inhibitory signals. So, you have to balance out depending upon the micro environment. So, that is, and also in addition, immune system is also there. Sometimes it protects. If it is a chronic inflammation, the immune system may help in progressing the tumors also, right. That is also another thing. The epithelial markers stain a cancer which metastasized into bone marrow. Here they have shown 2 micrometastatic regions of lung cancer are seen in a lymph node. So, by immunohistochemistry they may be able to identify, especially in the lymph node, they may be able to identify whether it is ready for metastasis. Usually people look for the nodal presence. For example, in breast cancer also, even after treatment or during treatment, they will look for the presence of these cells in the lymph node. If it has spread to lymph node, then there is a possibility of metastasis. So, that is one indicator, it is a very important indicator. Lymph node, presence of cells, cancer cells in the lymph node is an indicator for metastasis. So, that is what people generally look. Sometimes it may not be true, because the patients who survive 5 years after cancer therapy show few positive. That means, that is, if they really survive for a longer time, then you do not see lymph node, this one. If you see lot of them, then probably they will get metastatic. After 5 years, if you do not see that, then that means somehow the system has protected you and you do not see that, you do not see them. So, one of the earlier theories said that lymph node positivity is an intermediate step in metastasis. One idea was, if you have lymph node presence, they will always get distant metastasis, but this is not true, but this was an early view. New view is that cancer patients with more than or equal to 4 lymph nodes will test positive for tumor cells or at very high risk for distant metastasis. So, what it means is a via media theory. We can only say they are at high risk. We cannot 100% say that they will get metastasis. That is the new view. So, anyway, lymph node status is one of the most useful prognostic indicators in breast cancer. Current methods have seen 10 to 33% of women diagnosed with the negative lymph nodes also develop metastasis. It is not a complete 100% confirmatory test. That is what I just told you also. That is, the new view is that yes, if you find in lymph nodes, it is a high risk factor, but 100% you cannot say. That's what it is. Some methods were taken up, like frozen breast specimens were collected from women with negative and positive lymph node status, and people have isolated RNA and did microarrays. Data suggested that a molecular signature can you do with and without lymph nodes. Somehow they did not get very good results by this kind of analysis. So far whatever we have seen, the properties, anchorage independence, motility, properties of metastatic cells, invasiveness, and most of the cancers arise from, the metastatic cancers arise from epithelial tumors, and the epithelial form of tumor can temporarily show a fibroblastic form. So, one of the important components that is somewhat acting in between is the so called extracellular matrix. All of you know what are the extracellular matrix proteins: collagen, elastin, fibronectin, laminin, glycosaminoglycans, all these things. I hope all of you must have studied this. Integrins also very important because they connect the extracellular matrix with the cytoskeleton. They play a very important role in enforcing this anchorage dependence. Then another parameter, especially since we are going to talk about epithelial mesenchymal transition, the most important molecule that people look at to represent what is E-cadherin, anybody knows? I told you epithelial mesenchymal transition, which means epithelial cells are transformed into mesenchymal phenotype. So, E-cadherin will represent epithelial morphology. So, they interact with the catenins. Catenins connect the cadherins to the actin cytoskeleton. Integrins also play a role in this, transmitting the stimulus, mechanical stimulus from extracellular matrix to the cytoskeleton, anchorage of the cell motility, activation of RAS-MAP kinase pathways. These are all the important functions. Now, certain integrins can help in anchorage independent tumor cells to survive. You can see here in the left portion, when it loses anchorage, tumor cells lacking integrin expression, they die. That is, they will undergo apoptosis. This one, when it loses anchorage, the tumor cells expressing integrin can survive. So, that is what this happens because integrin can signal via SARC. What is that? CAS, anybody knows? I do not know whether I have told. There is a molecule, it is called P130 Cas. I also do not remember the full form of that, I forgot somehow. Suddenly it does not come to my mind. And then FAC is a pathway, SARC Cas FAC pathway. So, it turns on the, what you have to remember is that it can turn on the cell cycle. This is why under these conditions it can still survive. Then the Rho C it's called Rho factor, Rho C protein can make the cells more fibroblastic and metastatic. This is also very important. You can see some of the stained cells here for Rho C expression. Rho C is a signaling molecule. Okay. So, its expression, if it is weakly metastatic, you can select the variants which are highly metastatic also. By if you add the dominant negative rho C expression vector, then they become weakly metastatic. That means rho C contributes to this factor. Suppose you block this rho C, then they become weakly metastatic. So, that is the experiment they have done. So, you can look at it, they are weakly metastatic. If you express rho C into those cells, I hope you understand this by looking at the figure. That is, you take one by one, then you will understand. Suppose you take weakly metastatic cells, now introduce rho C into them by gene expression, then they become highly metastatic. Do the opposite, highly metastatic cells, you inhibit rho C by using a dominant negative construct, then they become weakly metastatic. So, which this experiment tells you that rho C contributes to metastatic phenomena. Similarly, SRC you know is a tyrosine kinase. Many of you got confusion. SRC is a receptor or non-receptor tyrosine kinase? Non-receptor tyrosine kinase. I remember some people writing receptor and all that. It is a non-receptor tyrosine kinase. Receptor tyrosine kinase examples are EGF receptor and all that. So, do not get confused, PDGF receptor, insulin receptor, those are the examples for receptor tyrosine kinases. Non-receptor tyrosine kinases are SRC, you can say JAK, FIN, S, LIN and all that. I remember, but you may not have noticed that. While we took those classes, we have mentioned that, but it may be slightly difficult for you to remember them. So, SRC transformed fibroblasts, they have done some experiments that they become invasive, which means just like the other experiment I told you, rho C contributes to metastasis, SRC also contributes to invasive phenomena. Now, we are talking about epithelial mesenchymal transition. This is very important. Epithelial mesenchymal transition is a genetically regulated process by which epithelial cells are converted into more fibroblastic mesenchymal cells. So, when I see what generally in practice in research what we do is, we look for E cadherin to represent the epithelial phenotype, N cadherin for mesenchymal, or there are other parameters, several transcription factors we can look at, which are important to identify the EMT. We are going to talk about them, that is why I am not telling. So, mostly it originates from epithelial tumors. So, we need to better understand the epithelial cells, and when the epithelial tumor becomes mesenchymal, it moves and invades. The ones they become mesenchymal, they acquire the property of invading. So, that is important, and also motility, movement, they can move and invade. So, this property is obviously essential for metastasis. Now, one of the most important inducers, although we have studied TGF beta signaling is a tumor suppressor pathway, right, but TGF beta is also a strong inducer of epithelial mesenchymal transition. This may look like a paradox, but under certain conditions TGF can induce metastasis. Here you see TGF beta treatment alone is sufficient to induce EMT. Mammary epithelial cells, RAS transformed cells, they show high expression of E cadherin, right, but if you treat them with TGF beta, they become metastatic. The other marker I told you is vimentin. Usually that is what we do in the lab also. E cadherin we will look for epithelial morphology. Mesenchymal morphology we will take either N cadherin or vimentin. Vimentin is the main marker for mesenchymal cells. This is a very important thing. It is reversible. EMT is reversible. What is the reverse phenomenon? Anybody knows? simple naming. If you call this as EMT, you call that as epithelial to mesenchymal. No, you call it as mesenchymal to epithelial transition. Do you understand? You put that EMT, and if you reverse it, mesenchymal to epithelial. I will tell you, there is an interesting thing in this that is written here. That is what the first sentence itself. See the question mark and all that. Actually, I must thank one of my, that time I requested a guest lecturer. Her name is Vanil Sumantran. She prepared these slides actually. Thanks to her, she has written all these things. She used to take class in those days. Now I am using her slides to teach you, that's all. Most metastatic cells isolated from tumors have epithelial morphology and behavior. If EMT occurred, why they have not appeared fibroblastic? Now I just described to you, if the mesenchymal morphology has come, then they will be fibroblastic. But then when you look at the cells, metastatic cells, they do not look like that. Then you are confused. Why? The answer to that is, it is reversible. So, what happens at a given time point you are looking at, it depends on that. So, it is reversible, it is transient, that is the most important. That is why you are not able to detect the EMT cells sometimes. That does not mean metastasis has not occurred. So, EMT, although it is a good marker, still it can be deceptive. It can cheat you. You are looking for EMT, you think it has not occurred, but it actually occurred and reversed. So, you are not able to detect it. So, that is what happens in immunohistochemistry sometimes. Mainly happens just before tumor cells move from primary to secondary site. So, EMT may be reversed after the tumor cells metastasized and formed colonies and adapt to the new site. So, that is why a controversy exists whether or not EMT actually takes place in vivo and whether EMT is really required for metastasis of solid tumors of epithelial. So, this is a controversial subject because of that. I have attached a PDF file, if you noticed, you please read that, those of you who are interested to read, because it is one of the latest reviews written by an expert, Sendurai Mani, who is actually he is a postdoc of Weinberg. I told you, Bob Weinberg is a very big person in whose lab the first human oncogene and first human tumor suppressor genes were discovered, RB and what is the other one? What is the first human oncogene discovered? Is it RAS or SARC? SARC or RAS? Are you sure? SARC. Chicken, it was discovered first, then they have discovered it in this one, right? SARC only is the first one, I think, right? Or RAS, human form is RAS. Chicken, yeah, human, you are right. The human oncogene is RAS. SARC was from chicken. That's what I am also telling. The first oncogene, if you just name the question, word the question as first oncogene, then it is SARC. But if you say human oncogene, then it is RAS. So, that is the small difference, and that was discovered in that. Now, this is also an important slide. So far, whatever I have told you about epithelial mesenchymal transition, we have given some kind of tabular form. That means, what are the important changes? There is loss of cytokeratin, E-cadherin, because E-cadherin is lost when you have EMT. You look for E-cadherin, but E-cadherin will be less if the cells have metastasized, that is the idea, EMT. Epithelial cell polarity, then it acquires fibroblast like shape, motility, invasiveness, mesenchymal gene expression program, Mesenchymal, that is what N-cadherin I told you. The another important thing that I did not tell you is MMPs, matrix metalloproteinases. MMP2 and MMP9 are important components. Vimentin, fibronectin secretion, PDGF receptor expression, alpha V beta 6 integrin expression. So, these are the important changes that occur during EMT. Not only that, we have some transcription factors also. I will talk about that shortly. The EMT induction can happen due to several factors. They are all given here. You can look at it. Several signaling pathways are involved, and finally, there are also lot of transcription factors which are involved. So, ultimately EMT facilitates invasion and metastasis. EMT facilitates intravasation of tumor cells and subsequent formation of distant metastasis. Here only I want to say the important transcription factors. These are very, very important, especially when you are studying EMT. SNILE and SLUG, they are transcriptional repressors of E-cadherin, which means they will promote or they will inhibit metastasis. They will promote. So, expression of SNILE and SLUG can induce EMT. So, EMT markers are SNILE and SLUG. It is very important markers. TWIST is another marker, may also induce EMT and is also implicated in the regulation of metastasis. So, these are the important ones. Why in particular RAS-MAP kinase has been shown to activate the RAS-MAP kinase pathway. If it is activated, that will activate SNILE and SLUG. RAS-MAP kinase pathway is for cellular proliferation, but it does not mean that all the time you will have the RAS-MAP kinase, the proliferation will eventually result in EMT. You cannot say that, but RAS-MAP kinase pathway many times is known to activate SNILE and SLUG. The other pathways are Wnt, SRC, ROS, beta-catenin, Notch, integrin. These pathways can induce EMT. So, this slide is very important because it tells you what induces EMT, what are the transcription factors that may be important in EMT. The other problem is, it is not, I told you it is reversible also. So, what makes it to progress further? That is the active stroma, which means the microenvironment. So, there are number of factors and cells also. You have fibroblasts, you have the immune cells, you have the cancer stem cells, like that you have lot of cells. You have the blood cells also, endothelial cells and so on. So, lot of these cells, they secrete several factors which ultimately can move this into metastasis or may even block this from becoming metastatic. So, that is what is important. So, the proliferating endothelial cells expressing the receptor for vasculoendothelial growth factor, these are important. Fragments of extracellular matrix molecules, broken parts of basement membrane, active proteases, matrix metalloproteases. This is another important thing I told you, tumor associated macrophages. So, the immune system, instead of protecting you, turns into a facilitator of metastasis under chronic inflammatory conditions, for example. Okay. So, tumor associated macrophages play an important role, and then as I told you, carcinoma activated fibroblasts, they are called CAFs. The fibroblasts are very important. The molecular mechanisms, because MMPs can activate, for example, can cleave the insulin like growth factor. MMPs can activate other growth factors like TGF alpha. MMPs can also promote the release of TGF beta and TNF alpha. MMPs can promote angiogenesis. MMPs can also regulate invasion and migration by degrading the ECM components such as laminin, laminin 5, fibronectin, agrican and so on. So, these are the different mechanisms by which ultimately you promote cell proliferation, angiogenesis and so on, and invasion. The calves, the so called fibroblasts, they can, for example, here they have given some examples here. What is that? Normal human prostate epithelium immortalized with the T antigen, these cells form small tumors in mice, but if you add the carcinoma activated fibroblasts, they promote the formation of very large tumors. That means here calf is helping to form more tumors. So, that is one example they have given. Similarly, there are reciprocal interactions between TAMs. What is TAM? Yes, tumor activated macrophages and tumor cells. So, in response to EGF, cancer cells secrete what is called colony stimulating factor, which can act on TAMs which have the colony, because why they act on TAMs? Because it has the receptor, colony stimulating factor receptor, CSF1 receptor. So, TAMs can secrete EGF which can stimulate proliferation. So, it is cancer cells provide colony stimulating factor for TAM and TAM provides EGF for cancer cells. So, like that it can. Another thing they have done, CSF production by carcinoma cells. Macrophages can stimulate motility and invasiveness. Yes, that is also they have proven through several experiments. Let us not worry too much about it. If you want, you can go through that. I think we are almost done. The primary tumors have small subset of cells which express genes that specifically favor metastasis. Can we identify some metastatic genes? So, these genes may function as some signature for metastasis, would be enriched in circulating tumor cells. That is why people are looking for circulating tumor cells and then see whether they have any metastatic associated genes. If you can take the tumor circulating cells, circulating tumor cells as they are called, CTCs, circulating tumor cells. So, you can look for metastatic genes in them. If you find, probably that will help you to understand that it is going to be metastatic. But then these are a little bit controversial because different tumor types have different ways of metastasis. That is the answer. And people are also looking for microarrays to identify a set of metastatic associated genes. That is, can you give a gene signature for metastasis? So, that is what many people are working. For example, they will take some metastatic tissues, say 9 down regulated genes, 8 up regulated genes, like that. People have studied that. People are actively looking for some specific set of signatures. That is also going on. A lot of such works are going on. And then see here one such publication. I think my colleague has taken some publications which show that a 5 gene signature as a potential predictor of metastasis in colorectal cancer. But these are kind of difficult to 100% say that they will predict metastasis. So, these people are claiming that the 5 gene signature may be able to predict metastasis and survival in colorectal cancer. Colorectal cancer is one cancer where people have worked a lot, especially in the US. Genes associated with increased metastasis, I have already told all these things: TGF beta, transcription factors, MMPs, only one more thing, it is called urokinase type plasminogen activator. UPA as it is called, angiogenic growth factors. So, these are the important factors. Hypoxia we have already discussed, how hypoxia is related to angiogenesis, all that. You read that, you will understand. If you have this hypoxia, then that leads to resistance to chemo and radiation therapy also. This part we have already discussed that, you can go through that. And I have also told you about the microenvironment, that is cancer associated fibroblasts, endothelial cells, pericytes, immune cells, and then cancer stem cells. So, it all depends on the interaction between these different cell types that will ultimately determine whether the cells have to go metastatic or not. In addition, we also have the cancer stem cells, which may also play an important role to see whether they have to go for metastasis or not. So, I think we will stop here.